My name is Claire Elwell. I'm one of the professors in medical physics, and I also have the honour of being today's speaker's PhD supervisor. It's an absolute pleasure to be here to welcome you all to the inaugural lecture of Professor Ilias Tachtsidis. And I want to extend a particularly warm welcome, not just to everybody in the room, but to everybody online as well. Thank you all for joining us here. For those of you in the room, we know that there is a selection of colleagues and students and friends and family. And it's particularly wonderful to see Ilias's family here, and particularly Ileana and Phoebe here in the front row, very proud of their amazing dad. And also to other members of Ilias's family who've actually travelled here all the way from Greece. So thank you very much for joining us. So an inaugural lecture is a big deal. It's not easy to become a professor at UCL. The promotions process really is seen as the pinnacle of an academic career, and it involves the assessment of research, teaching, institutional citizenship, and enterprise and engagement. These are all qualities which you're going to hear that Ilias has in abundance. But Ilias didn't just have to convince the UCL colleagues and promotions panel, he also had to achieve 6 references from other world-leading institutions. And having been a referee for other promotions, I know that the primary question that we take notice of is, would you promote this candidate at your institution? So Ilias's promotion to professor at UCL isn't just an acknowledgement of what UCL thinks of him, it's an acknowledgement of what several other world-leading institutes think of Ilias's work and achievements. So it really is an enormous achievement and a great honour, and very suitable that we recognise it in this way. So what we're going to do, we're going to have an introduction in a minute from Professor David Delpi, who, when I said, Dave, what do you do now? There's a pause because Dave does everything. Dave has been at UCL for over 40 years. He's now an emeritus professor at UCL. Also Ilias's PhD supervisor. And I would say the godfather of the technique of near-infrared spectroscopy, of which myself and Ilias have researched over the last few decades. So Dave will give an introduction, we'll then hear Ilias's lecture, and then I'll return at the end of the lecture for the vote of thanks. So again, thank you all for joining, and it's my pleasure now to introduce Professor David Delpi.
Welcome, everyone. It really is a great pleasure, a really great pleasure to be invited to introduce this inaugural lecture. One of the best things about being an academic is just seeing the students who you're working with just growing in their capabilities and their confidence. And this is even more so with your PhD students, given the quality of the PhD students that we can attract here at UCL. As a supervisor, you spend most of your life actually just hanging onto their coattails and trying to keep up with their progress and their innovation. It's a fantastic thing to do. It may seem like from the outside it's hard work. The hard work is from the supervisor just trying to keep up with what the students are doing. Ilias was an exemplar of this, and as you'll see, and hopefully from the slide, Ilias was always working. This is him in his earlier days. On the right, he's working with Terence, who's actually sitting in the second row, working on genuine science. But on the left, the two of them are doing equally valid work at a wine tasting session at an international conference, equally important and an equal part of the community of science, and a great place to actually find out what's really happening, because it's never in the papers that you read. It's always in the mind of the person that you're chatting to at the conference. If I had to identify a specific characteristic of Ilias that caught my attention, it would be his unfailing enthusiasm and energy. He was hardly ever down, and if he did get down because an experiment didn't work, he just bounced back the following day. The only thing I ever heard from Ilias was, yes, Dave, of course I can do that. Always. As a result of that enthusiasm and his abilities, he very quickly completed his PhD, and the rest is history, as you're going to hear now from him. So as I said at the outset, it is a real pleasure to invite Ilias, Professor Tachtsidis, to give his inaugural lecture. Ilias, over to you.
Thank you so much for these wonderful words. I would like to thank everyone for attending in person and online. I'm extremely happy to be here and celebrate my professorial promotion. I got my chair in biomedical engineering in 2021, but due to pandemic, we had to wait 2 years to organize this. So today I hope I can entertain you and educate you talking about the wandering photon. The photon that always tries to move further and further, predominantly through forward scattering. Thanks, Dave, for this excellent picture. And I'm going to be talking about a lot of pictures, and I'm going to show you a lot of pictures. So, being a professor, one must be wise beyond their years. And before we talk about science and engineering, I must thank my wonderful family, my partner, Nikki, and my 2 little ones, Phoebe, who is over there, 8 years old, and Iliana, 13 years old. I want to thank Nikki for 23 years of patience and being there after the long days, putting up with me and my working hours with minimum complaining, but with lots of shouting. I will also thank to my family that are listening now in Thessaloniki in Greece, my father and mom, who do not understand English. I hope someone translates them. My sister, who is also here, and with your family, also my uncles and my aunts and my cousins. Hopefully one thing I'm going to try to do with my lecture is try to explain to them what I have been doing in the UK for 26 years. And let's start our wandering photon science journey with this picture. This is 2003, 1 year into my PhD, at the meeting of the International Society of Oxygen Transport to Tissue in Rochester, USA. These are the 3 people that at that point of my life I was spending most of my time with. David Delpi and Claire Elwell, my supervisors. And by the way, I still remember Claire's surprise when I burst into her tiny office to let her know she would be my supervisor. So I broke the news to Claire. In the middle next to me is also Terence Young, who was a postdoc in the group at the time, who was sitting at the desk next to me and putting up with all my questions about near-infrared spectroscopy, near-infrared light, and also the person responsible for learning MATLAB. Terence, I still have all the scripts that we've done. I have them in my computer, and I do share them with my students. I have been extremely fortunate that I have Dave, Claire, and Terence in my life. Not a lot of things are said about staying close to your supervisors following your PhD. But in truth, having Claire and arm reach has been extremely valuable for me, not only from the academic part of things, but also from the life part of things. I have learned a lot of how things work, and thank you so much, Claire, for that. So thank you, Claire, for taking me as your PhD student and making me part of your family, and sharing this amazing journey in science and life. So the wandering photon story, importantly, also, beyond people, needs locations. So these are some of the places that I have done work in UCL from my PhD years until now. Indeed, what makes UCL a wonderful and magical place is the access to all these institutes and departments and hospitals and the people that come with them. I have also done work outside UCL, and I did spend some time in Germany, and I'm going to talk about that a little bit later on as a research visitor. And I have been very fortunate to have the funding to actually do this. So let's start our journey, and let's introduce the photon now by talking about the electromagnetic spectrum. I want to introduce the near-infrared photon, which is just beyond the visible light of the spectrum, just beyond the red color. The near-infrared photon is invisible to the eye, but has some amazing properties that David Clare introduced me in the early 2000s. So one of these properties is that while the visible light, such as the green and the blue, are completely absorbed by the tissue in such a way that your fingers in this picture look like a black shadow, and you can see them here, the red light manages to escape the tissue. And as you approach the infrared light, and using here a very specialized camera, you can see your fingers being transparent with the shadow of your blood vessels. That's amazing. So this is a better illustration of this. Again, this is my hand against the red light, the infrared light. You can kind of see the vessels through my finger there. So near-infrared light is transparent to bone. And this was described very well with a publication in 1977 by Francis Iopsis. And what I'm going to show you now is a little video that we have done with the team as part of our public engagement activities. And hopefully to show you how this was discovered, how Iopsis discovered this effect. And again, thanks to my PhD students for doing such a great work with this. So hopefully you can hear it.
Francis Iopsis' dinner featured a grilled chuck roast, an American cut of beef which contains part of the shoulder blade bone, much like the one Izzy has here. Iopsis and his younger son Paul cleaned muscle tissue from the bone before holding the pink object up against the light. They noticed that a diffuse red light could be seen passing through the bone. Iopsis realised that if visible red light was able to pass through, then near-infrared light with its longer, more penetrating wavelength should also be able to pass through the bone. He wondered if near-infrared light could be used to beam through human bones.
Iopsis had discovered a way to shine a light to illuminate the inner workings of the human body.
The next step was to take this bright idea and turn an optical phenomenon into a tool for medical research.
So one of the things that Francis Iopsis, how Francis Iopsis discovered that near-infrared light can go through bone is by having his lunch dinner. He had a nice steak, and then he found on the bone that near-infrared light actually can go through that bone. So this is us acting on that event with a nice piece of steak. And this is my PhD students, Josh and Izzy. And at the back, you can also see some of my other students. So this was our attempt to kind of demonstrate how that discovery was done. And this is a true story, right? So taking this into consideration, that the fact that near-infrared light can go deep into the tissue, and the well-known fact that the arterial blood is full of oxygen and bright red, we can now create fingerprints of oxygen levels in the blood deep into the tissue. So we can use these differences in light absorption, in light color, between the oxygenated and the deoxygenated hemoglobin, the main molecule in our blood that carries the oxygen, to quantify your tissue oxygen levels and our blood volume. So using two different colors of light, or two different wavelengths, we can separate the deoxygenated hemoglobin and quantify oxygenation. As our red blood cells give up oxygen, and as our red blood cells take up oxygen, they change color. And we can actually quantify this non-invasively and in real time. And one of the most important organs that needs oxygen to function is your brain and your neuron cells. So, the concept described here with this complicated figure is related to something called the neurovascular coupling. And with this, I'm going to demonstrate how we've been using light to monitor brain function. So your neurons on this side, they activate, they produce electrical activity, so you can talk and move. And that needs oxygen. And what happens is that when that process happens, oxygen is delivered to particular areas of the brain, and that causes an increase in the oxygenated hemoglobin and the decrease in deoxygenated hemoglobin. So we now have a way to monitor with our light the increase and decrease oxygen deoxyhemoglobin so we can quantify brain function. Another technique can also do this, called functional magnetic resonance imaging, or fMRI, but it's a much expensive technique to use and very difficult to operate. So let's demonstrate this with a great example that Joey Hertz from Yale, one of our sister labs at Yale, shared with me. So we have here our subject, and on top of the head in this cap, these black wires are fiber optics. There are fiber optics that send light onto the head, and the reflected light from the brain we receive. So if we look at a particular location of the brain as our healthy volunteer is performing a motor task, like a finger thumping, what we will see an increase in oxyhemoglobin and a decrease in the oxyhemoglobin when the person taps the fingers. It goes to rest, and come back again. So for me, it was extremely important to actually understand what's happening. So a large part of my work has been understanding the oxy and the oxyhemoglobin changes, and beyond that, ensuring that the measurements are reliable and true reflection of the brain activation. All this within the concept of having a portable, relatively inexpensive FNIS instrument. You can see me here with one of our large FNIS systems from Simanju, a Japanese company, and behind me is Paola Pinti, a postdoc, a team member, an expert in this technology, and now lab manager to an exciting new facility called the Toddler Lab, which we're going to talk a little bit about later on. Here, I would also like to take the opportunity and mention the great relationship that we have with Japanese industry, who have been pioneers in disseminating this technology to the market. To start our journey of understanding the physiological origin of the hemodynamic oxygenation changes, I was one of the first persons to integrate our brain FNIS measurements with our systemic physiology. And these are shown here. While everybody was concentrating on what's happening on the brain and trying to measure oxy and deoxy, I was interested to know what's happening in the body and how these signals could affect what's happening in the brain, and also how might actually interfere with our FNIS signals. So I used systems to measure the blood pressure, I used systems to measure the scalp blood flow, and so on. But what are these instruments? And in the early 2000s, this picture shows some of the instruments that we have been using and some of my very happy student volunteers. It's worth mentioning here the Nairo system from Hamamatsu, which is on the top, the Atini system here, the Hitachi TG100 system that is shown here, which was one of the first commercially available FNIRS optical topography system. I was a very happy 22-year-old student opening a box, arriving from Japan with that instrument in there. I was so lucky that Dave has made that arrangements. And I have to tell you that we still have that instrument. It works pretty well. It still measures what it's supposed to measure. And I'm very keen to try and find a place for it, to display it. So also around 2010, I had access to some of the top range systems, custom brain systems. This one is the brain imager system from PDB in Germany. And I'm going to talk a little bit about that later on. So now I have the instruments, we have the technology, we have the hypothesis. What is next? So let's start now engaging our brain. And I was trying to think, I wanted something very simple. I just want to create a brain function, and I want to see what's happening to the brain and the body. And I thought, let's start using anagrams. So what is an anagram? So icon is a four-word anagrams, it becomes coin, reserve becomes reverse. So that's the only thing that the volunteers had to do. And as they did that, I was monitoring their brain. And I observed something that looked like brain activation, so you can tick the box, but something is not right. The changes are too large, and the dynamics do not fit the theory. So red here is oxygenated hemoglobin, and blue is deoxygenated hemoglobin from two different subjects on the frontal part of their brains. So yes, that looks like activation, but is it? In fact, when we look closer to the systemic variables, in particular here in the blood pressure, I see very large systemic changes. So here the bottom graph showing the blood pressure from the two individuals. And I see also differences between the individuals. So to understand more of what's happening here, I needed to understand what is happening to the hemodynamic and blood volume changes, not only in the brain, but also in the scalp. So I needed to measure the blood volume changes in the scalp. And I thought, being at UCL, surrounded with all these magnets, I could find somebody to convince to do a measurement in the magnet of scalp blood flow. And the person saving me actually was not a UCL person, was Heidrun Wobnitz in PDB in Germany. She was very interested on my early findings, and she handed me over the keys to the lab, introduced me to Zhenya here, Kirina, and Alex here, Zeltsov. They gave me the keys, and we started putting people in the magnet, not to measure the brain activity, but to measure their scalp blood activity. And we're one of the first people, and one of my kind of very famous kind of discoveries, demonstrated that I get a blood oxygenation signal on the scalp, on the vessels on the scalp. We now have an issue. And that issue is that this signal now can interfere with our FNIRS images of the brain. We're very happy to see the community nowadays understanding that this is an issue. I need to remind you that it was back in 2010. Still, people didn't understand exactly where the signals are coming from. The manufacturers nowadays acknowledging that this is an issue, and with the help with colleagues, such as Felix Coleman here, we push to suggest solutions both in hardware and software to solve the problem. We now have tools that can help with this and make our measurements more reliable, and everything, in my view, started from those simple anagram experiments that I have done with the help of students in the department in the early 2000s. So where we are now, as our FNIRS technology has been moving outside the lab to ecological, naturalistic settings, I recognize the need to investigate and develop robust methods and protocols to allow this translation and make our measurements reliable. And for that, I needed the help of our neuroscientist colleagues at the Institute of Cognitive Neuroscience. So with help of Paula Pinti and Antonia Hamilton and Paul Bergis, we set up the FNIRS Cognitive Neuroscience Group. And at that point, I was very pleased to meet Joy Hers, that is shown here. So Joy was another neuroscientist at Yale, and it was interesting to monitor two brains synchronously, something called hyperscanning. So she approached me because she knew that our technology can do this. So we've been working hard as well, collaborating to try to achieve hyperscanning with FNIRS. And just recently, my PhD student, Uz Hakim, shown here, Uz had demonstrated how we can scan one brain here in the UK in UCL and one brain over the Atlantic at USA in Yale, simultaneously as people interacting. Again, demonstrating the strength of this technology. But what about the next thing about near-infrared? So I've got another video here.
At the moment, they do this using 2 or 3 specific colours from the light spectrum called near-infrared. We want to go further. By using the full spectrum of red and infrared wavelengths, we can get a much richer, more detailed view of the processes inside the body. We can also measure how oxygen is being used to produce energy inside cells. This is possible because these light wavelengths are able to detect an amazing process called metabolism. Metabolism takes place within the mitochondria. Mitochondria are miniature energy factories inside every cell in our bodies. If we can detect the colour changes within mitochondria in the brain, we can see how well our brains are working. So how do we shine light through the bones of the skull into the brain tissue and then detect it coming out the other side?
This is a little video that we also did again with the team, trying to explain about broadband news. While most of the FNIRS technologies out there commercially available, they only use the 2 wavelengths, we want to use the full spectrum. So we want to use the full spectrum of light to look inside the brain. And the reason that I wanted to do that is that we wanted to see if we can identify other contrasts, other optical contrasts beyond the hemoglobins, beyond the blood. We were very interested in particular on the mitochondria. And we knew from our friends, the biochemists, that the mitochondria, the electrotransport chain or the mitochondria, the enzymes called cytochrome, they do have optical contrast. And one of the things that we're trying to do with my team is trying to develop this further. So what we target now with near-infrared spectroscopy, what it now defined as broadband is the mitochondria and not the blood vessels. In fact, the blood vessels and the blood hemodynamics are an issue and a confounding factor. There are particular molecules within the mitochondria, we call them cytochromes, that facilitate energy production, and as they do, they change color, as I said. So, while measuring hemodynamics, we only need 2 wavelengths. For measuring cytochrome and metabolism, we need to extend our technologies and we need to measure multiple wavelengths, hundreds of wavelengths. So by that we can measure now not only what is happening to the blood vessels, but what is happening inside the mitochondria. So we can quantify something called the cytochrome C oxidase. We can do this with optical spectroscopy, something that the team at UCL have been working since the 1990s. At that point, resident biochemist Chris Cooper, shown here in this picture, was heading that work. And Claire managed to convince Chris to collaborate in 2004, 2005. Now, I'm not sure who convinced who, but Chris was back. But I was the engineer tasked to make this happen. So how you go about pleasing physicists, medics, and biochemists that you can measure cytochrome C oxidase. As an engineer, I start building instruments. So I need to resolve the issue of reliable measurements of mitochondria function. And I'm going to build instruments to do that. So I need to use the latest technology, and I need to improve what we measure. So here I'm showing a bit of a timeline of all the different systems that we've been building at UCL since the mid-90s, and then when I kind of started working on this around 2000. But if we're talking about instruments, let's have a look at them. So there are quite a few of them, but that's what I do. I build instruments. We start with the old technology, and indeed you can see here the CRT monitors and the old desktops. And we actually integrated our systemic measurements with these instruments in our endeavor to understand what we measure with broadband news and metabolism. I will show later on measurements from those early studies. In 2008, I built a new instrument that is shown here. I call it FOSS. It's a unique hybrid instrument that integrates broadband ears, integrates two technologies and frequency domain ears. It was the most reliable instrument that we had, but it was very complex and difficult to operate. And definitely I knew that would not be used for clinical purposes. Later on, I was joined with PhD students, and with Gemma Bale, we built Cyril in 2014, and this is the instrument here, with the purpose to take this system into the neonatal intensive care unit. And Izzy here, Izzy the rover, Fong Fan, and Frederick Lanz, we built more instruments. With Izzy, we built the Fan Cyril, which is a multi-channel system, and I'm going to talk a little bit about that later on. So we wanted systems with better technologies, more imaging capacity, and more reliable and user-friendly. We didn't stop there. We also made them smaller, and that was what my PhD student Pardis Kainas had developed. We call those systems mini-Cyril. We achieved that by making connections, direct connections to the industry, and talking directly with the manufacturers, adapting and modifying what they were actually producing. You need to understand that none of these companies were making components for us. They were making components for astronomers and biologists. We also had to be more adventurous with our technology, and I would join with Luke Toon, a PhD student, to develop a multi-wavelength time domain system, an instrument, a near-infrared instrument, unique, that could measure more than 16 wavelengths, the time of flight of photons. An instrument that my postdoc Frederick Lanz later on called Maestros, and we are currently even updating. So I'm very sad that I cannot actually be the engineer updating this system, so I'm very jealous of Fred, that he has all these new components, very expensive ones, building a very unique instrument at the moment. And we also collaborated with colleagues from ICFO Spain, and particularly Turgut Teruran, where we try to implement and integrate other optical technologies like diffusion correlation spectroscopy to also measure blood flow and to actually achieve the true potential of optical technologies. And this is Florence 2018. In addition, with my PhD student Lucas Giannoni, we actually developed a hyperspectral near-infrared system shown here. The reason for that system was to try and do broadband microscopy and trying to actually resolve cytochrome signals from the cortex. In fact, that particular instrument was the instrument that helped us support an EU grant that I'm going to talk a bit later. So we have built the instruments, how we can use them. We need now to use them to demonstrate that we measure metabolism. So Martin Smith, the neuroanesthetist at the Hospital of Neurology and Neurosurgery, said to Claire, Claire, you can come in the hospital, you can use my office that I do my consultations, you can use that little office next to the intensive care unit, said to Ilias to bring the instruments, and we can start doing some clinical studies. So these studies start first by hooking ourselves, so this is me, by the way, so hooking ourselves into our instruments So this is me and Martin Tisdale, a neurosurgeon. Me and Martin Tisdale spent hours and days in that small office next to the intensive care unit, making people hypoxic, giving them CO2 to breathe, lots of CO2, and also taking blood samples. Of course, we're very happy to do that on ourselves as well, as you can see here. Everything through due diligence and ethics. No adverse effects. While in the morning we did this in healthy volunteer studies, in the night we would go and use these instruments to image traumatic brain injury patients. And this is one of our instruments in the neurointensive care unit on a traumatic brain injury patient. Martin Smith made sure to remind us every morning in the ward rounds how important it is to do these measurements of brain oxygenation and metabolism in these patients. And every time we'll ask us about our measurements. So I went through 2 neurosurgeons, Martin Tisdale and Anab Ghosh, and 1 neuroanesthetist, David Delpi. All of them successfully finished their PhDs. And I said to them at the end, you also get a full engineering qualification. We also had some medics skipping out on us because it's difficult. It's difficult to do this study. But with the perseverance of Martin Smith and Claire, we have for the first time understood what we measure. And let me show you what that is. So this is a study on a healthy volunteer. And what we've done is that we reduced the oxygen levels down to 80%. Your normal oxygen saturation as measured by pulse oximetry should be around 97, 100%. And we actually give them nitrogen to breathe for several minutes, and we took their oxygenation to about sort of 80%. So this is the type of oxygenation you get in your blood if you go in very high altitudes. We're one of the first people that actually did these measurements because we wanted to see what we can measure with near-infrared on the brain. Important, what we measure in terms of metabolism. So we can see for the first time the brain oxygenation dropping, following these hypoxic events. And importantly, we can also see our metabolic cytochrome signal here also being affected. We found out hypoxic thresholds of metabolic dysfunction, and in the meantime, we demonstrated to the EF-NIS community the physiological changes in the hemodynamic signals for the first time. We did more than that. We increased brain blood flow by breathing CO2, and we did cause some minor headaches in the process. We increased the oxygen levels by giving people oxygen to breathe. But what else we could do? Can we do some further engineering to understand our measurements even better? The ethics did not allow us to induce cardiac arrest and resuscitation, which would be the last kind of thing that we wanted to do. So, as engineers, we have to develop models. For that, we have to develop a computational model of physiology. So that work started with Murad Banaji in 2000, a brilliant mathematician that built the first model. And this was part of my PhD, in fact. This was one of the main aims of my PhD. It took him a couple of years, and our first publication of our first model was 60 pages long. The reviewers took them another 2 years to review that. The modeling work was a major part of my PhD in understanding the brain oxygenation measurements. And me and Murad spent endless time studying physiology and biology. With Chris Cooper shown up there, every now and then popping in to remind us how little we knew about the mitochondria, and upon reflection, how little everyone knew. The model is a computational representation of the physiological processes in the brain tissue, from the blood vessels to the mitochondria, a sophisticated way to understand what your measurements represent. This was important for me as I got my first fellowship in early 2010, and I wanted to move this technology to the neonatal clinical unit, and importantly, to start measuring newborn babies. To achieve this, I recruited our PhD students. So Tracy Morris was my first PhD student working on the model, then Tharindi Hapwaraki, a second PhD student working on the model, and my latest PhD student was Josh Russell-Buckland, working also on the model. And by the way, Josh is actually coming back after some years in the industry to join the team. We also had a computer scientist, Matthew Caldwell, that joined us as well. And the work that we did with Matthew with the modeling is to try to understand neurovascular coupling and the confounding effects of FNIRS. And of course, David Delpi, that I'm showing here as well, an sociologist, looking at using the model to predict and quantify impaired brain autoregulation. We've been doing this for some time now. And beyond the beautiful maths, this is how the model looks like. It's a messy network of nodes, all of them interacting with each other. And understanding these interactions allow us to understand the mechanism behind our measurements. For me, what was the most important thing is that we could predict when our measurements were wrong. And beyond that, using the model, we can also predict when there was a brain injury through our measurements and the use of the model. So that takes me now to my next type of work, which was translating broadband news in the neonatal care. And in 2010 and 2015, I secured 2 Wellcome Trust fellowships to allow me to do this and establish a true multidisciplinary team of physicists, engineers, and clinicians at UCLH. UCL Medical Physics and UCLH Neonatology, they have a collaboration of more than 40 years, a collaboration that started with Dave and then continued with Claire and others. So I was very lucky to be able to continue with this collaboration. We have something very unique across the world. That collaboration makes UCL and UCLH top in translational research that nobody else can achieve. Through these initial steps, I started working with Subha Mitra, we saw here in the picture, as one of his PhD supervisors. And as you will see later on, and we discuss some of our results, I'm very happy that Subha actually has a faculty position at UCL, so we can now continue with our collaboration and with some exciting new projects. So why the baby? So why the newborn baby? There is definitely an urgent need. There is an urgent need for real-time, non-invasive measurements of the brain tissue oxygen levels and the biochemistry of the baby to assess how well the baby is doing and try to develop effective neuroprotection strategies, such as hypothermia at birth asphyxiated infants. These infants, during birth or in utero, they had an episode of asphyxia or what is called hypoxic ischemia. So the blood flow and the oxygen to the brain was reduced. So it's important that we be able to diagnose these patients, these babies are the caught site. But first, how I can take my broadband system into the clinic, I need to convince the clinical team that what I measure is real. So how I would do that? With the help of physicists, and in particular Alan Baybridge, we start taking my instrumentation into a 9 Tesla MRI magnet to do metabolic spectroscopy. We did proton MRI spectroscopy that allows to measure lactate over here, which is a byproduct of metabolism. We also did phosphorus magnetic resonance spectroscopy with that very expensive 9 Tesla magnet that allows to measure the energy, the ATP levels into the brain. And we've done that on the piglet, which is the preclinical model of the baby. What I wanted to do, I wanted to demonstrate that we can do these metabolic measurements with my broadband ears, and there's no need for a magnet. Of course, I didn't tell them that at this point. But let's see some of our first results that give me confidence. And this is some of our 2 studies from 2 different piglets. At this stage, there is a decrease in the oxygenation levels and the blood flow to the brain. And we see my gray signal, my broadband cytochrome signal being reduced, and the lactate with the MRS, the magnetic resonance imaging going up. And of course, in this particular piglet, they never returned back. But in this piglet, following the ischemia episode, we can see the lactate going back to normal and my cytochrome signal overshooting and then slowly going back to normal. This was one of the first studies that we saw that our measurements of cytochrome with the very inexpensive broadband systems follows the lactate measurements that they've done with proton magnetic resonance spectroscopy. We did more. Here, for example, we did measurements with the phosphorus coil looking at the ATP levels and trying to see how my green signal here, which is the cytochrome signal, relates to the ATP levels, shown here with this purple signal. And again, we identified that in some piglets, our signal, the cytochrome signal shown in green here, did not recover, the same with the ATP. In more than 30 animals, we demonstrated that our broadband measurements of cytochrome can predict the ATP recovery. And importantly for me, they can predict piglet survivability at 24 and 48 hours. And all that with 30 minutes measurements 30 minutes of measurement just following of the insult. And this is with a system that is a fraction of the cost of the MR. So here, for example, every point is a piglet. And you can see a very nice threshold between the piglets that did recover the insult and the ones that did not recover the insult. But what about the babies? So this was our motivation. So our focus was the newborn baby brain and neurodevelopment conditions. So we want to take our instrument into the hospital, into the neonatal intensive care unit to help our clinical colleagues and support our patients. So Suba and Gemma, around 2013, 2014, start setting up our instruments in the neonatal unit. And these are some of the first pictures that we took of that. So this is one of our instruments next to the baby cot side. This is the baby over here. And what we've done, we're actually trying to do a very sophisticated setup that we only not measure what is happening to the brain, but also trying to measure everything that is going on with that baby. And our task was to try to do that measurement within the first days of life. So I was very excited when Suba started collecting the first data sets. And I remember when he called me to talk about this particular baby, one of the first babies that we monitored was a 10 baby, almost 41 weeks old, and was admitted with clinical seizures. At the start, when I was having a discussion with him, I think very early in the morning, we thought the system was not functioning properly because the system was giving two measurements, one on the left side of the head of the baby and one on the right side of the head of the baby, and they were very different measurements. So we looked at closely on this, and we plot here the brain oxygen levels and the brain cytochrome levels. And we found that there is different relationship between the left side of the brain and the right side of the brain of this baby. In fact, from our modeling, we knew that this linear relationship between oxygenation and metabolism does not look right to me. We know that this was unhealthy. And 5 days later, when the baby could actually move into the MRI, we did observe a stroke on the left side of the baby's brain. A very rare condition for a 10 baby, very difficult to diagnose. But I was so pleased that our technique could do that from day one. I think Suba was pleased as well about that. But we continue with this, and with Gemma, we did more babies and further work. We followed again the same concept of analysis, of looking at the relationship between oxygen and metabolism. And here I give you again two more examples of two different babies. So I'm plotting down here the oxygenation of the brain and the cytochrome levels on the brain. On one baby here, you see not a lot of great relationships between the two. And another one here that we see quite a big sort of high slope relationship between the two. So this was a baby that had a moderate injury. We actually discovered that at around day 7 doing our normal biomarker of lactate levels. And this one had a severe injury, again, identified around day 5. So by using our broad-bandage measurements of oxygenation and metabolism, we can identify very early on with a very high sensitivity, a specificity, which babies they're going to have a severe injury, and they might actually have very significant motor problems or even death. With Suba, we even went further, and we looked at this, looking at the relationship between the blood pressure that we measured on these babies and the broad-bandage measurements of metabolism. So on the top of the red trace is the blood pressure of the baby. The green one is our broad-bandage measurements of metabolism. And we actually developed some tools to look at how these signals are co-vary together. So here is this marker. So the more black areas and the more red areas, the more worse for the babies. So the more red areas and the more black areas means these two signals, the blood pressure, the metabolism, are going together. So this was actually a baby that had a moderate injury. But if we look at another one here that had a severe injury, you can see larger now black areas showing very good correlation between the blood pressure on the top and the metabolism, and again, more red areas. So you can look at this again. And Suba followed these babies until they were 2 years old. And we did these measurements within the first days of life. And we could actually relate these measurements to how the baby were doing when they're 2 years old, with a very high sensitivity and specificity. So I was very happy with these developments. And I think also our clinical teams were very happy. So what else are we doing? These results will allow us to push our technologies even further And just before the pandemic, we managed to establish our Florence system. So this system was used to monitor both now term and preterm infants. And this is work that also Kelly, Harvey Jones have been doing with the support of Frederick. They monitor a lot of infants. We have now more than 80 babies that we monitor with an instrument that measures cytochrome, metabolism, oxygenation, and blood flow as well. This is a unique instrument that only a couple of labs in the world have. But we're moving also further forward, trying to introduce metabolic imaging with our instruments. So not just try to image the oxygenation, but also the cytochrome, also the metabolism. So you can now get these beautiful maps of hemodynamic activity, but also metabolic activity. And this is work that we've done in adults and also babies, work that led by Paolo Pinti and also Mahin Siddiqui, that he has a welcome fellowship to continue with this. So we have now established a very unique but fraction of the cost of the typical MRI system imaging way to look both on brain oxygenation and metabolism. And we have now our latest system that is here, that Fred developed, is now moved to the toddler lab with what we want to do is to image the brain of the toddlers. It's a very new, exciting lab that just started, focused just in toddlers. And you can see very happy faces of people just having this new instrument in that lab. But we also build a lot of instruments and give to a lot of people. So our world domination of broadband needs has just started. So we give an instruments to Sydney Hospital, to Justin in pediatric cardiac surgery. And by the way, I do believe, and I still am going to say it for whoever listens there, that the cytochrome is best suited for this type of application. And I'm really looking forward to see some of those results from cardiac surgery. We send instruments to Brisbane Hospital to look at anesthesiology. Instruments to University Hospital of Psychiatry in Zurich, again to look at psychiatric disorders. There is a very significant link between psychiatric disorders and mitochondrial function. We send an instrument to Columbia University, Department of Psychiatry, again to look at psychiatric disorders and mood disorders. And also we develop and send an instrument to Neurology University of Lübeck in Germany to look at Parkinson's disorders. And I don't have time, but we still develop more systems, so there's more things and more applications that I'm hoping that we can do in the future. But what about our future developments? So with SUBA, we just secured major funding to expand our technologies to placenta and fetal monitoring. This was funded by the Wellcome Leap, a very big collaboration that not only includes UCLH, but also includes a hospital in India. We're very excited about this. This started in October, but we're moving very quickly ahead. One of the key engineering challenges for us is to build a wearable broadband instrument, to build a new optical sensing technology, if you like, and method that we can image the placenta and we can image in depth more than 5 centimeters. And the key to develop this is these new technologies that I have access, in particular these miniaturized spectrometers that I show you here. So with my PhD student Musa Talati and our technicians Temis and Iwiki, we're trying to build these instruments. And I'm looking forward to see where that will take us in the future. And what next? We also started a new grant to translate our hyperspectral imaging system that we developed 2 years ago with my PhD student Luca Giannoni to image glioblastoma tumors during open brain surgery. And for the first time, we want to use the broadband cytochrome measurement as a biomarker to identify tumor borders. This started in October. It will last 5 years, a collaboration with colleagues from Italy and Florence University, a Technical University of Munich in Germany, and also France University of Lyon. But what about new sensors and new technologies? Can we detect photons from the center of the adult brain? For that, we need something new. We need to look at different sensors, and that perhaps can be given to us through superconducting nanowires. In April, I'm off to Netherlands to kick off this new EU grant that we have, a collaboration with colleagues from Politecnico di Milano, ICFO from Spain, and Delft University in the Netherlands. Very exciting times for in vivo optical imaging, and I'm very happy that at UCL we can actually have these technologies with us. But the wandering photon should be more and beyond the science and the engineering. So public engagement is an important activity, in particular for scientists and engineers that are interested in developing technologies to help the society, and in my case, to support my clinical colleagues and improve patient outcomes. The clinical needs are simply an engineering challenge, and that's what I always say to my clinical colleagues. So public engagement. I was very lucky to be introduced to this by Claire, who can be seen here, by the way. And this is me, by the way, as well. And you can see some of the other faces, and Dave is over here as well. Right, so this is Sending Light to the Human Body, the Royal Society Summer Science Exhibition in 2006. Science is fun, Claire told us. And yes, it was fun and hard work. Lots and lots of hard work. But for me, it was the first time that I could see myself getting outside the lab and outside the hospital and talking with people enthusiastically about my technology. And I did like that a lot. And I've learned through this a lot. So it was my turn. And indeed, when I started my fellowship with the team, we tried to secure funding to do more of this. And we created our public engagement alliance called Metabolic. So this was funded by the Wellcome Trust, a collaboration between engineers, physicists and mathematicians, clinicians and doctors, nurses. And what we wanted to do, we wanted to shout to the people about all these new developments that we're doing and all these new instruments that we're trying to implement in the hospital to help our medics and support our doctors. We call this Engineering Light to Monitor Brain Health in Babies. We have attended more than 20 events. We have done a lot of resources, magazines and films, a lot of student placements, school visits. I cannot even count them. One of my favorite ones is the Royal Academy of Engineering Lightsaver magazine. They call it the Lightsaver. I quite like that name. Very successfully was actually given across the schools. And I said we've done a lot of festivals, but this is a short video that we've done after the Big Bang Fair Festival in Birmingham. It was very good to talk with all the little ones that were interested in our technology and interested in engineering and science. And we did give a lot of gummy bears. And you can use gummy bears to explain optical absorption. And this takes me to the next step. So it was now my time and my turn in 2019 to lead, with the support of Suba and the team, and of course the little ones, you can see them here. This is Phoebe and Iliana. A Royal Society Summer Science Exhibition in 2019. So 13 years after Claire introduced me to this event. So it was our time now to do it. We were so popular that the Royal Society gave us an extra room just for us to create a virtual neonatal intensive care unit that is shown here. We can also spot Dave over here trying our near-infrared hand scanner, and my PhD student Josh trying to explain near-infrared to Dave. Or maybe the other way around. It was a great event. The whole team joined, and this was the last soiree, all the people. So this is me here, Suba, Gemma, Fred, Susie, Luca, and of course our nurse as well. What you can actually do as well with these events is that you do get your 5 minutes of fame, and we had a lot of different ones. I mean, I just put a couple of pictures here. The one that was perhaps the most significant for me was this one, the evening standard one. Infrared device called Serial can reduce risk of cerebral palsy. So this was done on the evening standard, so it was read all the people that were taking the tube at the trains that day. And it did. I know that they read it because the next day they came to us with this paper saying to us, explain us what you're doing. So definitely that worked. And now we do even more public engagement, coming out of the pandemic. We have a very good project called Orbits that we're trying to go to schools to talk about technology and make young engineers and young scientists. So as we're coming to the end, I must remind everyone that I'm also an academic. And since my PhD, I never stopped doing teaching and supervising students. And there is a fact here. I did all this even though UCL first time paid my salary in January 2021. I think that puts a lot in perspective. So this is some of pictures of me with my postgraduate students as part of our yearly poster event. Everybody looks happy. It was just after the exams and they haven't got their marks yet. That's why they're happy. And one of my most proudest moments is when I get these emails out of the blue from my students thanking me about teaching them optics and how to code, and then telling me how important that has been in their job careers and the job they're doing now. But it's extremely difficult to balance research, academic duties, and family life, in particular without support. Getting now to my conclusions, I need first to say apologies to the people that I have not mentioned, to the collaborators that I have not mentioned. So this is the list of my co-authors from my 180 publications and counting. I have need at least 12 hours to go through everything and everybody. So I concentrate mostly in the early work that I've done. So yes, I have done 180 publications and I have secured more than 50 million of funding across the different UKRI charities. But this is only the standard metrics and standard tick box. How about you quantify the public engagement activities and the student supervision, how you can quantify what these are producing. I believe it's an exciting time to be involved in near-infrared technology and optics. There are new components out there, there are new technologies out there, a lot of appetite to move outside the typical laboratory clinical testing to ecological settings and to the home. And our technology can do this. Our technology is inexpensive. It can be at the court side, at the patient side, is pain-free and can save hospital staff time. The research output from our field, as you can see here, is increasing exponentially, but we're still some way to go. But other technologies like the fMRI, you can see that for several years now they have plateaued in their output. So definitely it's an exciting time to be involved in NIRS and functional infrared spectroscopy. Of course I will show more pictures. So thank you, Claire, for your support in this journey, for making me part of the family, introducing me to all these wonderful people. I want to thank also Martin for teaching me the importance of working together with engineers and clinicians. This is Martin and me. So this is me, by the way, playing football. You see that here. This is Claire washing the dishes, by the way, in front of a lot of empty bottles of wine. Thanks also to Chris Cooper over here, for reminding me every time I see him that I'm not a biochemist. Yes, I'm not a biochemist. Also thanks to the team. And you can see here Fred getting his award, and Gemma over here. I'm very excited with Gemma that just started her lab in Cambridge. And Paula here that is doing some exciting work at the Total Lab. It's definitely a lot of exciting times. And my final concluding slide is just to say how exciting it is to be involved in NIRS nowadays. And if you're a student out there, if you are a researcher that you're trying to find a science family to join, I would say this is the best time to come and join us. Come and join the near-infrared spectroscopy research field, a very exciting field with a lot of future. Thank you.
Well, Ilias, well done. And we can see how much work went into producing this talk, but actually how much work you've done in the last 30 years. I've known Ilias, as you've heard, for a long time, and I've always, as Dave said, has seen him as someone who's hugely resilient and always busy. And I think you've seen now what he's been busy with. So an extraordinary talk and a summary of an extraordinary career and lots of things that you've done that you haven't had time to speak about today. Let's just give Ilias another round of applause. It's very well deserved. So it's my job now to give the vote of thanks. And really, I'm going to give that thanks on behalf of a range of different people. The first is I want to say thank you to Ilias for all of those who've benefited from his research. And as we've seen, this includes a wide spread of applications in a range of challenging clinical environments across the entire lifespan, literally from measurements in fetuses all the way through neonates, toddlers, and into adults. So really there is not an area of brain research, it feels, that Ilias hasn't at least attempted and had an impact on. The other thing I think it's worth saying is that Ilias does the hard stuff. And it was interesting that Chris and Martin and I could sit and come up with ideas with Ilias in grant applications, and maybe we'd get them funded. But it was actually up to Ilias to build the systems that we decided needed to be built. And the interesting thing, I think, what you've seen from Ilias's talk today is that the engineering innovations that Ilias has put together are to understand the things that we really need to know, but are difficult to measure. And that's why Ilias is brilliant, because he doesn't shirk away from those challenges. And we need people like Ilias that take on the challenge of doing the hard stuff. So thank you, Ilias, on behalf of all the people that have benefited from that research. I'd also like to say thank you on behalf of everyone who's benefited from being a collaborator of Ilias. As you've seen, every single slide that he's shown us encompasses a range of interdisciplinary collaborations. And one of the skills that Ilias has demonstrated in his career is the ability to communicate with a range of different disciplines and to think about the language that's going to work best to get the job done. So thank you from all of the people that have benefited from the collaborations. I'd also like to say a very special thank you for everyone who's benefited from your teaching and supervision. You are one of the strongest advocates for the student experience that I know, Ilias, and you are hugely dedicated to both undergraduate and postgraduate student supervision. And as you've said, even when you were working on a full-time fellowship, you never left behind the duties of really providing first-class education. So thank you on behalf of all of those students. I'd like to thank Ilias on behalf of everyone who's benefited from his principles. For any of you that have been in a meeting room with Ilias, you're familiar with the situation where you realise that Ilias isn't happy with the way that things are going. And that's because Ilias wants to do the right thing, even when this may not be universally popular, and particularly when it's not always the easiest thing to do. I admire your principles, Ilias, and thank you on behalf of those that have benefited from them. I'd like to thank Ilias for those people who have benefited from his public engagement activities, which I'm so pleased to see included in the talk today. You've had a huge impact on the next generation of engineers, Ilias, and you've also used these activities to bring together your team. You look at the people who've contributed to those events, many of them in the room here today, and you can see how much enjoyment there has been to gain from those activities, even through what I know that you understand as being a lot of hard work, and work actually that is often done out of hours and on a volunteer basis. So thank you. There will be people now that have seen your exhibits, that have heard you speak, that will be doing engineering because of that event in their lives. Thank you on behalf of them. Thank you on behalf of everyone that has benefited from your support of EDI, and especially Ilias, your encouragement of women in STEM. And I have seen this firsthand on many, many occasions. You are a very strong advocate, not just paying lip service to what needs to be done, but you're one of the best allies I can think of really changing the face of physics and engineering. And just, you can see from the people that have worked with you and what they're now going on to do, really benefiting from your support and encouragement in a very practical way. Thank you very much on behalf of all the women in STEM. Thank you, Ilias, from me, a personal thanks, for being a superb PhD student. What a blessing to have a PhD student like Ilias, Dave, wasn't it? For being an inspirational colleague, somebody who just was always excited about the challenge, and for being a true friend through everything that life has thrown at us. So for everyone here, we are going to have a well-deserved drink in Ilias's honour following this event. For those of you online, I encourage you to raise a glass this evening to toast Ilias's success. And also, don't forget that one of the purposes of this event is to connect people. You've had the pleasure of learning about Ilias's research, so please do follow up with him after this event. There is, as you can tell, there's no question that Ilias isn't prepared at least to attempt to answer. So I encourage you to follow up with Ilias with any research questions that you have. So all that remains to be said is, it's a brilliant lecture from a brilliant academic for a very well-deserved professorial chair at UCL. Well done, Ilias, and we look forward to celebrating with you this evening. Well done. All right, we can have a drink.
We can have a drink now.
Well done. Well done. Really good.